My brain hasn't come up with it. Usually, sometimes you like.
Now I want to know.
Yeah, yeah. Gosh. I don't think it was a side effect of the thalidomide. I just cannot remember it. It'll come to me. Trying to read this. Okay, that pause was my signal to jump in, okay? Well, here we are, Tuesday, September the 6th. And I got, some announcements to make, and then we'll go on to our material. But I sent an email, I think this weekend sometime, try to give you a heads up. The lab we're gonna do online, and there's a couple of reasons, there's actually 3 reasons. I thought we were going to do I knew we weren't gonna do a wet lab, but I got to look at those videos and I thought, at the moment, I can't add beyond that without showing you real things. So we're doing online lab tomorrow whenever you go there. Just to refresh your memory, here, you go there, and then I've got this note here about the lab. Online lab this week, that means tomorrow. We're gonna call that lab 2, but I'll tell you right now, we're not gonna do a lab report number 2, okay? So you're not gonna do a lab report on this one and get that out of the way right away. But you go to this website and just try to peruse or look through the rats, the cats, and the pigs. You don't have to do any further because I haven't got that there yet. And just see how the people did it. To be honest, it's nothing spectacular. And so, in one sense, that's really good because you can do something better. So, do that here. Well, so there's 3 things that actually reason why we're not meeting tomorrow face to face. There's that. But I don't, there's, how many where's the TAs? Raise your hand. There's a few TAs in here, yeah. Some of you know this and you saw me opening up all the drawers. The last time we had lab, we were opening up all the drawers in our 245, it's 245, isn't it? 1040? It's 1045, right? Yeah, that room, and there's nothing in the drawers. So that's a little scary when you have these animals to dissect and there's nothing in the room. I just got back from going through all the drawers in the middle room where it says prep. There's very few things there. And that's what I'm running into because I know, this is only the 2nd semester this lab was taught, but it's the 1st semester with me and everybody does things different. I'm not sure exactly what they did last time, but I'm more of a hands-on. That's why I ordered the pregnant cats, pregnant rats, pigs. But by next Wednesday, a week from tomorrow, I've got some scalpel handles myself, my personal ones. If you're familiar with scalpel handles, there's a handle number 3 and a handle number 4, and they take different blades. So I got some blades ordered, but we just don't have enough supplies for everybody to get into an animal, to be honest. Because we're gonna be very invasive. We're taking all the parts out, we're weighing the cat, we're weighing the rat, we're weighing the pigs, we're measuring. You're going to dissect out the whole gastrointestinal tract, hopefully from the esophagus all the way down to the anal opening, and dissecting such, you stretch it out as one pipe. That's very impressive to do. It's hard to do because everything's kind of coiled and it's got a lot of connective tissue, but you get to appreciate things like that. We're going to be looking for organs that the video doesn't show. To be honest, they didn't do all that good a job. So what you should do is watch those videos on the cat, rat, and pig, and write down some things that should be done different, because when you're doing that, remember how we work in partners? One partner can be doing things with their gloves on, the other partner can be taking pictures and videos with their phone. Because that's what you got to get used to. Take videos and pictures of things, and then you can share them with your partner, and then you can put them in your lab report. Because you should think of making a lab report, and I said this for number 1, but we'll get to that in a minute. But for all the other lab reports, you should do it such that you're going to teach a high school senior in biology or a freshman in college. And most of the videos don't do a good enough job. They're just showing you kind of superficial things. So I'm gathering things, and I'm glad I didn't, say we're going to dissect right away, because we should have lab paper that's plastic coated on the bottom. So if things get messy, then we just roll that up, and we don't have to worry much about the table. there's 2 little bottles of hand soap in there. I don't know if you've seen that little dial. No, I've got some professional grade stuff coming, because here's one rule you should have when you do wet labs with me. Before you leave, after you put everything away, obviously, but before you leave, you wash your hands good with this stuff that I've professional stuff I'm ordering, and then you wash your hands again before you eat. Okay, so this is kind of the lab. It depends on when you have the lab, but sometimes you might be going to lunch right away. But we're going to do everything with a little finesse. We're going to show you the right way. We're never going to hurry. And so here's the kicker. Next week, a week from tomorrow, which would be, tomorrow's the 7th, so it's the 14th, right? The 14th is the next Wednesday. Make sure you come that we're going to do a wet lab. Now, if you don't have a lab coat, I've got some like plastic aprons that aren't thick, but they're cheap, and we have plenty of them. And then see if you can bring safety glasses. I've got some, but some of you have some chemistry, you know, a friend, you can have some, and I'll have some. So I think we should be okay. But what we're going to do next Wednesday is there's going to be a team of people do the cat. They're going to open the cat up. Another team does the rat, one each lab. So one cat per lab, so that's 4 cats in a row, and one pregnant rat for each lab, so that's 4 rats, and then 1 pig. And I always want to say fetal pig, but remember I told you I don't know if they're 2 days away from birth or 2 days after birth, and we might never know, because if an animal has taken a breath, you can float the lungs. But these things have been sitting in liquid, that fixative, that may not be a valid test. So I'm not sure if we're going to be able to tell if they were born or not. But there'll be some people doing the cutting carefully, but there will also be other people doing a little video or an image. Like we're going to try to, like today I'm talking about adrenal glands. We're going to try to find the adrenal glands of those things. And that's not easy to do because they're always tiny by the kidney, because AD, remember, adrenal, AD is near the renal gland. So there's things like that, and as each week goes by, we'll get more invasive, the more supplies will come. But I can't take off too fast because all the drawers are empty. So now I'm trying to find a place where I can put some of my own personal stuff, like for show and tell. One day, I'll bring a human uterus. Well, I don't want to leave that any place. I don't want to lose that. I'll be bringing big bore testes, but it'll be one of the show and tell and learn, okay? Anyway, we're going to get there, and I'm excited about the pregnant ones. I hope the cats have a variety of pregnancies, you know, like they're 63 days of gestation. I hope they're all a little older than midpoint, because then I can show you how to sex a fetus and things like that. So anyway, so that's, any questions about tomorrow? You're going to do it on your own. You're going to watch the videos, and some of the supplies haven't come. That's another reason. And here's another thing that you'll be doing instead of the lab. So I'm back on this page now, our main page, and I'm trying to figure out where should I put these 2 practice paragraphs. I guess right here underneath here, because this says reading videos for the week. So I'm going to make more space after dissection guide, but above this line, and in here there's going to be 2 practice paragraphs for the exam that's coming up. Okay, so it'll say practice paragraph 1, practice paragraph 2, because you're going to be doing, I haven't quite decided, 4 or 5 per test. Okay, and the test is already due, I think it says 9:15 someplace in Brightspace. But you're going to do, let's say, 5 paragraphs in Word, because I'll have a Word document that you can upload that says, you know, create a paragraph, a physiological paragraph that uses these 10 items, whatever, there'll be the 10 items. But when you submit it, please make it into a PDF file. And you can do that different ways through Word. And then you're going to upload it as one document. You know, it's going to be, let's say it's five paragraphs, then that's 5 pages. Okay? So do that, and by noon tomorrow, those 2 paragraphs will be linked right here under the rat dissection, but above this line. Okay? So then you can work on that. Because, and if that's up there by Wednesday, on Thursday you can ask questions about it. Because don't be surprised if there's some words or items in there that, like, what in the world is he thinking? Okay? And the other thing is, remember, the tests and all the lab reports are supposed to incorporate everything we do. For whatever we read, So there's really three sources of information. Whatever's on this page, whatever I say here, and whatever we do in lab. And the paragraph on the test should incorporate everything. Maybe there'll be a prefix IPSI in the test. What would one of the first things you would think about making out of that prefix? How to use it? Ipsilateral, right? That's not the only thing you can do with IPSI. And you can find out all the things you can do with IPSI, right? But it's one of the obvious ones. Or like a number, 45. How are you going to use 45 in a paragraph? Pack cell volume. How would you use 100 in a paragraph? Lifespan of the red blood cell. Things like that. So they're going to look for you to incorporate things. If you just use 100, like, let's say we had 100 dogs and, you talk about 100 dogs. Well, is that physiology? It might work out good. I don't know. I'm just saying everything should be related to the physiology that we did or beyond. If you know physiology beyond this class, you could incorporate it. That wouldn't bother me at all. But don't go below. Okay? Just trying to give you a little strategy. So then I talked about the lab. So now I'm going to jump up here, lab report number 1, guidelines. I'm going to click on that. That was last week, basically anatomical terms, and this will show you the title and a few things to think about. Guidelines for lab report number 1, and you're going to upload it as lab report number 1. But notice underneath there, does not apply to any other lab. Because all the other labs are going to be more the histology, gross anatomy, physiology, pathophysiology, and then a 5th section that you decide on. Like maybe a personal story that you have related to kidneys or atresia ani or something, depending on the topic. So here it is, anatomical terms. The report should be a combination of paragraphs, lists, and images, right? You took a picture of the cat. You could explain the label. What does a plain cat mean? You know what I mean? What does that plain on the label mean? You could list all the terms and then write a paragraph about them, because that would be actually informative for you to write a little paragraph about anatomical planes and just make sure there's a list. Say, here's the list that I just talked about, or you could start it at the beginning. Here's a list of anatomical planes, now I'm going to explain them. Or here's an image I found off the internet that I like about horses, and it shows all the anatomical planes. So use your imagination, but make it such that you show what you've learned and other people could learn from it. And so basically that means written in such a way that a reader of the lab report would learn the presented material. Not just a reporting of what you did, but a reporting of how to learn this stuff. So the sections talk about rods, show and tell and learn. That's how I started the lab. That's how I'll probably start most labs. Then we did anatomical planes. Don't forget oblique. Directional terms, rostral. Proximal. And then there's some other, general terms, like, I guess that's where I would be thinking about the ipsilateral stuff. Fossa, foramen, maybe things you want to add. It says discuss 12 other anatomical terms. Maybe you could say, we didn't do this in lab, but I want to talk about disarticulation. Somebody can do disarticulation in the cat if they wanted to, but you'd have to look it up and know what that means. And then a section of your choice. Something about maybe something personal, like, yeah, I worked someplace and we often dissected the animal down the medial plane to look for something. A lot of places that have a lot of animals, they examine why the animal died, right there. Chickens, pigs, a lot of animals. It's hard to take animals away someplace to have them looked at, because you never know what they died from, right? You don't want to take things around. Anybody have any questions on that lab report? Notice I don't have a number of pages minimum, but it's just, something that would be adequately covered. So I hate to say number of pages. You know what I mean? That gets to be grade school-ish. Make it look like you're in college. Maybe that's the best thing I can say. Anybody have questions on that? Okay. So then we're going to talk about vasopressin and adrenal, but I want to go back to one thing about the renal system down here that I didn't talk about really, and it's some more hormone stuff about kidneys. Okay, so that's where I'm going to take off, and I'm trying to promote that people read these things and glean from it, and hopefully you don't need me to hold your hand and take you through things how to learn. You know, you're not a freshman, right? And I think I made that point before. It's hard to predict what you need to know in the future. If we knew that, then we could say, oh, you don't need to know anything about hormones or whatever. It's amazing how at least half the people in here, if they predict a career, that's not what they'll be doing. You know that quite a bit. I guess this is someplace, got some advertisement that's not going to bother me. Okay, Labor Day savings, I don't want to buy anything. So I'm fighting this. So when we talk about renal function, it's amazing that the kidney makes hormones and sends them out, but also gets affected by hormones. And you know a hormone is like a key, and it's looking for the lock that it fits into. And then if it turns and something happens, then that might be called the parent hormone or the native hormone. But then if Eli Lilly synthesizes something that fits in there and turns it and causes an effect, then you might call that an agonist, A-G-O-N-I-S-T. I'm not sure if I ever said that before. Agonist. Agonist. An agonist is a compound that acts like the native hormone. So if I say we're going to inject this progesterone agonist into the horse, it's going to have a similar effect to progesterone. And then when you talk about all these hormones, I don't think, I might have said it once, they all live a certain time in the animal, and that's called the half-life. That's half, H-A-L-F, dash life. And it's a very important concept, because depending on the half-life, tells you how long that hormone is going to act within the animal. And sometime maybe we'll get into how do you give animals hormones. I can make an implant of testosterone and put it under the skin of some animal, and it'll give that animal testosterone for a long time. But if I inject some hormone IM, there's going to be some kind of peak value, then it's going to drop, because it's going to be degraded. Everything is degraded, and that's called half-life. So let's say, and we might get into that, it's in some of the reading, but let's say the half-life of a hormone is 30 minutes. That means if you put in 10 milligrams, in 30 minutes there'll be 5 left, half. But the problem is, and you got to make sure you understand this, that in another 30 minutes there won't be 0 left. Okay, the half-life is 30, so you inject 10, and in 30 minutes there's 5 left, but then in another 30 minutes there's 2.5 left. And then there's 1 point whatever is left. You have it, you keep having it. It's not like 2 half-lives and then gone, no, and that's hard to understand. But somebody way back when, our ancestors said, we're going to talk about half-lives. How long does it take for half of it to be metabolized? But then we in the field say 10 half-lives equals gone. So we say maybe theoretically it's never gone, but in 10 half-lives. So in 300 minutes it's gone, if the half-life is 30 minutes, okay? And that half-life is used for radioactivity too. Like if you have, like we used to do hormone analysis with radioactivity, and I'm I'm trying to think of, I think the one compound we used, the half-life was 20 days. So in 20 days, it would be have half the radioactivity of when we started. Well, lo and behold, if we kept it for 200 days, then theoretically there's no radioactivity left. And Purdue used that same system. They would come and collect our waste, radioactive waste, but then if something was a 20-day half-life, in a year they would just throw it in the regular garbage. You can see how they would do that, right? It would decay down to nothing. But these hormones have a half-life, some have long, and you know if it's testosterone, and this key represents testosterone, testosterone looks like this the world around. Testosterone only has one identity, one picture. You know, it'd be the same ID for everybody that's known as testosterone. But if this was a protein-like hormone, then it varies by species, especially those protein hormones that are fit together by amino acids. The vasopressin, I think, has 9 amino acids, but it varies by species. So steroid hormones are constant the world around, and protein hormones vary by species. Sometimes they're very similar. Anyway, hormones are made by organs, and hormones affect organs. So we're talking about here, notice we've done blood, then we've done kidney because the kidney filters blood, and now we're just doing more things about kidney. Okay, so one of our links that I put up there was vasopressin, but lo and behold, our ancestors also call it ADH, and that's anti-diuretic hormone. And both names are pretty good, but anti-diuretic means the animal is going to retain more urine, more water. So make less urine, retain more water, see how that works? And the urine then tends to get concentrated, more yellowish if you have anti-diuretic hormone. If you give an animal a diuretic, that tells the animal make more urine, lose more water, right? So you can have either effect. And remember, we talk about for physiology, there's always a balance. Everything wants to be in some normal range. Okay, so they say are produced outside the kidney. Well, I'll tell you right now, they're produced, these two hormones, the same name, they're produced in what's called the posterior pituitary. Now, remember how for quadrupeds, we tend not to use anterior and posterior, but we don't, it's not pure. And so there's a anterior pituitary and a posterior pituitary. It's at the base of the brain. I've taken them out of a cow. You have to decap the skull. It's hard because you need a bandsaw. And then you can get the pituitary out. And the posterior pituitary is all neural. It's neural tissue. It's nerves. And nerves are releasing a hormone that goes into the blood. So if you wanted to do that study, you would call it neural endocrinology. N-E-U-R-O, endocrinology. Sometime if I have time, I'll tell you, when I went to a meeting one time called psychoneuroendocrinology. Very interesting. It was all psychiatrists and me. But I get along pretty good. We go to happy hour and we're telling stories, but it was psychoneuroendocrinology. Anyway, so the posterior pituitary, which is neural, makes these hormones, or I say these, but they're the same one, ADH equals vasopressin, and it goes to the kidney via the blood, because that's how all hormones are transported distances. If the base of the brain is making some hormone and needs to get to the kidney, that's like at least 4 feet away for a horse. It can't do that through what's called interstitial fluid. Interstitial fluid is the fluid that surrounds cells, and some hormones work like that. They're released by one cell, and they diffuse to another nearby cell in the interstitial fluid and never go to the blood. But if you're sending something from the base of the brain to the kidney, that's got to travel by the blood. There's nothing else that will transport it that far. So it's going to go by the blood. they're called chemical messengers, right? That's a hormone. And then remember, anytime you need to ask a question, you pop up your hand, and I'm stopping because, I mean, I can't tell you how many great questions we had last week in this class. Okay, so then here it is. Others are produced within the kidney. So now they're talking about kidneys that are hormones that are made in the kidney, and those have a local effect. That means when they diffuse to nearby tissue. That's called a local effect. So I'm trying to help you with some of the jargon. Okay, diuresis. That means, you know, you're making urine. So there's anti-diuresis, there's diuresis. Okay, ADH regulates water excretion. So now remember those three terms we had for with the function of the nephron? There was filtration, reabsorption, and secretion. The sum total of that is what gets excreted. So that's like a fourth term. It's not related to either of those other three, but the sum total of those other three make the urine that gets excreted. So there's a lot of things that can happen to the urine. Remember the arrows coming in, going out. And remember how I use the word reabsorption? I think some of the reading links sometimes say absorption, but you know why you want to correct that. You want to do reabsorption. Okay, so water excretion, okay, and then, you know, by permeability of the collecting duct. So what that basically means is that that collecting duct that was way off to the right in some of those pictures, that means water is leaving the lumen of the collecting duct and going out to the capillaries that are surrounding the nephron. Remember that picture where the nephron has got all kinds of blood supply around it? That's what this is saying. Okay, and then receptors at the base of the brain form the feedback mechanism. So whenever you see feedback mechanism, that's like something telling you from a distance what's going on. Okay? And then the feedback then either stimulates ADH output. Notice if the osmotic concentration of extracellular fluid is high. So osmosis, if you've forgotten that from biology, my definition of osmosis is the diffusion of water. It's the movement of water from high concentrations to lower concentrations. Okay? Okay, the receptors at the base of the brain. Yeah. And so the brain is complicated. It's going to monitor these things so as to concentrate urine. So if you concentrate urine, you're putting out less fluid in the urine. It's getting concentrated. Hormones of the adrenal cortex. So now that's another reason I wanted you to read this, because remember, the adrenal cortex is nearby. Hopefully we can find some in some of the animals, and the reason I say hopefully is because it's rather small. It might blend right into the fat tissue that's around, but if we're aware of it, we could do a little blunt probing and try to find it. We may not always be successful, but the other thing when we are dissecting animals, at certain times we're going to say, okay, well, I'll point to a group and I'll say, you walk around the room and see what everybody else is doing. And then when somebody comes by you, if you have something remarkable to tell them, like, oh, we found an adrenal gland, here it is, or look at the pregnant uterus, it had 8 cats. And you'll do that for every station, and then another group will walk around, because we're going to share everything we do as far as the dissection. And remember, one of the pair doesn't have to have gloves on. They can be doing the pictures and the videos, because that should be in the report. But so here it is, the adrenal cortex must release hormones. And I've got a secret to tell you. The adrenal cortex, all the hormones it releases are steroids. That means the cow adrenal cortex releases the same hormones as your adrenal cortex. And if one of those hormones are in low supply, you can give it to the animal and bring it back up. Remember how if you had a choice between having a low amount of something and a high, go for the low, because then you can supplement. If something's high, that's a devil to work with. Now you can do antibody treatment, but everything is temporary. It's terrible to have something too high. Anyway, in stress situations, okay, often the adrenal cortex, and I could say the whole adrenal gland releases material when there's stress. And we know about stress. I mean, it's amazing. Mental stress. You could be under stress either in a situation you're at, like a dangerous, if you hear gunfire, you're under the table and that's stress, but maybe the test that you're going to take determines if you graduate or not. That's pretty stressful. And so stress is a very general term, but the whole adrenal gland has something to do with stress. Not only, but even the medulla can release things when you're stressed. So let me see this picture here, if it enlarges. So anytime something enlarges, and I'm not sure if I can make it bigger. Maybe I can't, that's okay. Anyway, if things get bigger, view full size, here we go, okay, I should read that. So there's something good and something bad about this picture on the left. First of all, notice the kidneys are not exactly across from each other. There's always one that wants to be a little further ahead, more cranial. And you should be able to verify that on the cat, the rat, and the pig. See if you can. Then the other thing is, this depicts humans, and in humans, the adrenal glands are pretty much on top, the cap of the kidney. Here's the right kidney, and here's the adrenal gland. That's not going to be the case. Every animal I've ever dissected, the adrenal gland has been away from the kidney a little bit. And it's hard to find. And one of the reading shows you in a buffalo, but it ends up being cattle are the same. But in every species, the adrenal gland is a different shape. I know in pigs, it's elongated. It's like this, and almost like a little hot dog. But in cattle, one of them at least tends to be round like a disc. And if we find one in lab, I'll show you how to verify that you do have the adrenal gland, because that's another thing, to think you have it, but not to have it, right? And whenever you have adrenal gland, you can make a section, like a transverse section through the length, and you should see two regions, the outer cortex and then the intermedulla. And they're often a different color, because the cortex is pure endocrine, and the medulla is pure neural. So now look at that. I've just talked about two organs that had a pure endocrine function and a pure neural function. Interesting. So anyway, these are the regions. The adrenal gland has a capsule, keeps everything in shape. Then you have these three regions here, and each region makes a different hormone. But they're all steroids. And then when you get down here, the medulla, it looks different. So basically, by histology, when you do a lab report that includes the adrenal, then you could have a picture, histological picture that shows the different regions. And so the medulla, this is neural down here, and this is all endocrine. So this is all steroids. And these are, you would say, neuropeptides. Anytime a neural tissue releases something, you can call it a neurohormone. If it's peptide in nature, you call it a neuropeptide. And so that's it. Notice how, and now let's see if I can get out of here. Okay, well, here's another picture of it. This is the whole adrenal gland. And this cut here is enlarged over here, okay? So that's the cortex, this is the medulla, but there's obviously a blood supply, because you always have to have a blood supply in capillaries and venules. And so they called it adrenal cortex, they call it adrenal medulla, and when you cut that, you can see the difference. And we'll see on our tissue if we can, because I know in a fresh animal that I dissect, that just died, definitely the color difference is there. Now with this fixative, we'll see, maybe it drains everything, all the color out, and we won't be able to see the difference. Ideally, you do, especially if the animal. And then of course, this is showing the rest of the urinary tract. The ureters are there someplace, I'm not going to worry about it. Now I'm wondering if I can get out of there. I guess I can. Okay. Am I gonna do that? Sorry. Let's see. Okay. So there's that. That's what I did there. Hormones of the adrenal cortex. They're not only with stress, but they're important for renal function. And what you should do is list hormones that you come up to and tell what they do at the kidney. Do they make more urine, make less urine? Something. They always have some effect, and this would say it. In stress situations, as after an injury or a car accident, I had some friends that had a car accident, they were south of Indy, and they got rear-ended, and then they got pushed into another car, so their car got damaged on both ends. That's a bummer. So after an injury, trauma, surgical operation, hydrocortisone is a very common one. But it's a steroid. So hydrocortisone in you is the same in the bull elephant in Africa. Okay, and then this is a family name. When you see corticosteroids, that's like the Johnsons. And then there's individual names under the Johnsons, right? Bill Johnson, Jane Johnson, they all do different things. So you gotta remember, sometimes you'll see family names. Okay, and then is increased, so these hormones are increased when the adrenals are stimulated by. So whenever you see that, that says this is coming from someplace else and it's going in the blood to the kidney and stimulating it. And think of, you'll never know anybody else that's injected ACTH into heifers to see what the adrenal gland put out. That was me. So what you do is ACTH listed down here is adrenal corticotropin, and it's a hormone from the anterior pituitary gland. So there's a lot of chatting going on between tissues. So ACTH is made by the anterior pituitary, or I can buy a bottle of it and see what it does in the cow. This is endocrinology. Okay, a secretion. Now see, they said pituitary gland. In most cases in our written stuff, you should be either talking about the anterior pituitary or the posterior pituitary. Don't say something like ACTH is made by the pituitary. That's too general. It's made by the anterior pituitary. Okay, so it tells what it does. Output, urine, lack, okay, reduces the power of the kidney to deal with water loads. Hormone also promotes, yeah. Aldosterone, this is another hormone coming from the adrenal gland. And I'm just looking for some key words. I'm not trying to look at everything here that you're going to be reading. But remember, if you take notes, you just read it once, take your notes, and then never go back to this thing, if you've drawn out the name of the hormones and stuff. Now, here's another name of a gland. It's called the parathyroid gland. That reminds me of the paralumbar fossa. Para, beside. So, beside the thyroid gland maybe I should say, there are these parathyroid glands, often embedded in the tissue of the thyroid gland. And of course, have you guys been listening to the news? The thyroid gland in Ukraine, the nuclear reactor that might be in trouble. They're passing out iodine tablets. Anybody heard that? Did you hear, listen to that? You guys are too busy studying. Anyway, they're passing out iodine tablets. When you live near a reactor that might rupture, it spews out radioactive iodine. And it's the craziest thing. I don't know the physics of that. You know, how do you get radioactive iodine when this nuclear reactor bursts? And it's not the only one. But the danger about radioactive iodine is that your thyroid loves iodine. But the thyroid doesn't know if it's radioactive or not. And if it sucks up a lot of radioactive iodine, it'll destroy your thyroid. Thyroid glands would be destroyed by radioactive iodine. So what you do is you hand everybody iodine tablets. It's actually Ki, potassium iodide, and you fill up your thyroid with non-radioactive iodine. So then when you get exposed to the radioactive cloud that comes by, the thyroid will see the radioactive stuff but says, I'm full already, sorry, there's no seats for you. All the seats are filled by iodine. And the radioactive iodine then won't stick anyplace else in the body because the thyroid's is the tissue in the body that wants it the most.
What's the withdrawal time for an iodine tablet?
Oh, what's the are you saying half-life or?
Or how long would it take before you'd have to take another?
Oh, yeah. So the question is, how often would you have to take it? Well, of course, it depends on your body weight. You take it orally, and there's going to be a protocol. I don't know the exact protocol, but you'll take more than one, because it probably take, somebody has done the work where they've determined how much iodine a person that weighs like 150 pounds would need. I just don't know that off my top of my head. It varies a little bit. Okay, so parathyroid glands are next to the thyroid. They're important in calcium. I'm going to go on. Functions to excess. Look at this. If you have a tumor, sometimes tumors make an excess. And remember, I said excess is bad. It'd be better to have a deficiency. And so that's where these, and then here's the other thing about tumors. Tumors have a brain of their own, and they can make almost anything they want to. And that's why when somebody has a tumor, you might do some blood tests and you'll find out things like, and that doesn't make sense, and then one of the first things you think about is, I wonder if you have a tumor making this stuff that shouldn't be here in the first place. Tumors have a mind of their own, and they have all the DNA, they have all the instructions for everything your body makes. It's the weirdest thing. Do you know some tumors will make teeth? Some tumors will grow teeth in the middle of the tumor, and it could be in your abdominal cavity. The weirdest things happen. Okay, this goes on and on. Parathyroid, you know that. Vitamin D is interesting because vitamin D has been misnamed. You should write down vitamin D is a hormone. It's like a key that fits into a receptor. It's a hormone. And see, this is our ancestors. They mislabeled it. They called it a vitamin, and then later on they found out that it wasn't a vitamin because your body makes vitamin D. It's the sunlight on your skin. That molecule gets not totally activated, but in your skin, the first step of making vitamin D occurs. Then it goes to the liver and gets tinkered a little more. And then it goes to the kidney, and the kidney makes vitamin D, active vitamin D. Who would have thought, okay? The sex hormones, okay, now I don't really like to call them sex hormones, but a lot of places do. And what you should know is every sex hormone is found in each sex. So that's why I don't like to call them that. But because of the enzymes in females, in a lot of females, estrogen and progesterone are called the sex hormones. But males also make estrogen and progesterone, but they sometimes convert it to something else. And it's fascinating, and I probably will show you a chart on what's called steroidogenesis. And making different steroids is one enzymatic step. Okay, so let's see, okay, this is something in the kidney. If you see that JGA, that's the kidney, that's in the kidney, so look, it's near the glomerulus. Okay, here it goes, and renin. Renin is interesting, I'll let you read this, but some places call renin a hormone. It's not really, it's an enzyme. R-E-N-I-N, and then you should put on a sidebar, there's also R-E-N-N-I-N that you can run into, pronounced the same way. Okay, then you got this angiotensin thing. Whenever you see that, angio is a prefix meaning blood vessel, and tension means tightening. So that molecule must tighten blood vessels, must constrict blood vessels. Things are quite complicated. I'm going to quite get through this. Remember, I'm open to any questions today or tomorrow, but then when you see this word prostaglandin, that's an interesting name. And when it was first discovered, they thought the prostate made it. See the name? Prosta. They were trying to blame it on the prostate gland. Ends up being it's made a lot of different places. These are not steroids. These are not protein hormones. They're made from fatty acids. So it's like a third class, but most of them are called local hormones. Notice how I'm using that term. Most of them are made and then act nearby. And if you're into cattle, the uterus makes PGF2 alpha. That's capital P, capital G, capital F, and then 2 alpha. So the cow's uterus makes prostaglandin. It does, in this case, send it to the blood vessel, but it ends up going to the ovary without going to the heart, and it causes luteolysis. L-U-T-E-O, lysis. We had lysis last week, didn't we? Breakup of? Can't remember. L-Y-S-I-S means you're breaking up something. Anyway, hormone-like fatty acids, I just said that. So you can, I mean, it's pretty straightforward. Look at that. Unlike true hormones, prostaglandins are not transported away because they tend to diffuse through the interstitial space and affect another cell or tissue. But in my case of the cow, that stuff does go to the blood supply. But there's a special, and it's always got PG, prostaglandin, and I was saying then F, and then 2 alpha as a subscript. So these are different, but you should know there's probably 50 of these hormones out there. It's confusing really fast. Okay, so now they're talking about renal prostaglandins. That means prostaglandins made inside the kidney. Whatever, I'm going to keep moving on just to help you. There's PGF2 alpha. Okay, that's how you, oh wait a minute, no, it doesn't have the alpha, but PGF2 and then alpha is that's the Greek symbol alpha. Okay, vasodilator, that means blood vessels enlarging in their diameter. Vasodilation, vasoconstriction. Up and down. Why? It's not like pipes in your house that never vary. All these pipes, especially the veins, vary because if you're losing blood, they constrict and help you. If you got too much water intake, they expand to hold some of that extra water. Okay, but it's a pretty good article. They talk about enzymes, and it can get kind of crazy, but don't let it, you know, you start learning basic things, don't get buried down into the garbage. Okay, so here's another thing, the heart. We know it pumps blood, but you know what? It's an endocrine organ. It makes a hormone. In the tissue, it makes hormones, releases it into the blood, and that hormone goes to the kidney. And it tells the kidney to make more urine. Let's see if the paragraph says that. The most recently identified hormone that influences renal function is secreted by special stretch receptor cells in the atria of the heart. That's the smaller chambers. In response to more pressure in those chambers, the heart releases this hormone called atriopeptin, atrionatriuretic peptide, ANP, but it's also called atriopeptin, A-T-R-I-O-P-E-P-I-N. Atriopeptin is made of amino acids, so it's a protein-like hormone. So the kicker is the heart makes a hormone that tells the kidney to make more urine. If it feels like, man, there's too much blood here, and if there's too much blood, there's too much fluid, and the kidney will help you get rid of that fluid. So I can say ANP is a diuretic. So you put that together. Why is it a diuretic? A diuretic says make more urine. An anti-diuretic says make less urine. And it's always the kidney doing that, but it gets signals from different places. So, wow, look at how much to digest. Let's see, okay. And of course, any word that ends in urea means something in the urine. Glucoseuria means there's too much glucose in the urine. And remember, glucose should be reabsorbed, right? Remember in those diagrams? And then of course, edema. Edema is a collection of excess interstitial fluid. And I hate to pick on elderly women, but elderly women sometimes collect fluid in their ankles. Have you ever seen that? I mean, if they stand too much, men do too, but I think somehow it happens more in women. And if you actually could ask permission to put your finger on that ankle of hers, the impression of your finger would stay there when you removed it. And that also happens in dogs. And whenever you push your finger on some swelling and you remove it and it's still showing, that's called pitting edema. Pitting, pitting edema. It's got so much fluid that you press it and that spreads out some of the fluid in there, but you take your finger away, it takes a while for it to spring back. Pitting edema. Oh, I mean, look at this, all this stuff. Okay, it goes on and on and on. I want to go to something else. Anybody have questions on it? Yes?
Does the edema only happen in the ankles, or can it happen like in the upper joints or something?
Any place. Edema can happen any place. So the definition is excess interstitial fluid, period. So, this is extracellular fluid. It's, out of the vascular system, but it's bathing cells, and that's called interstitial fluid. Maybe I'll spell that for those of you that are like me, that aren't the best spellers. INTER, ST, STIT, is it? Interstitial, here it is, interstitial fluid, okay? Fluid found in the spaces around cells. So it's not in the blood vessels, it's not in cells, it's the fluid outside of cells. And there's always a balance, and if you get too much, that's called edema. Okay, so anyway, yeah. And remember, blood vessels, especially capillaries, are leaky. There's a lot of fluid moving out of capillaries, but then there's a lot of fluid moving back. But if you have more moving out than back, that causes edema. Okay? New interstitial fluids made, and you should know every fluid in the body is constantly being refreshed over time. Okay? Now I want to go to, let's see, I'll do a little on vasopressin and then I'll do the adrenal gland before the end here. Okay, so vasopressin, also called antidiuretic hormone, ADH, and it might have some other names here, arginine, vasopressin. It's too bad we can't just stick with one, sorry. Hormone made, okay, yeah. Now this you might say, I thought Rod said that vasopressin is released by the posterior pituitary, and notice it says it's made in the hypothalamus. So I have to tell you about that. At the base of the brain, there's a tissue that's all neural, pure neural, and it's called the hypothalamus. But it has neurons that send axons down to the posterior pituitary. So vasopressin is synthesized in the hypothalamus. Then it rides down the axon and then gets released by the posterior pituitary. So don't be fooled by that, because anything released by the posterior pituitary is made up in the hypothalamus, in the cell bodies of the neurons. And it travels down the axon, inside the axon, to get released at the posterior pituitary. So that's where that is. And you get these things called prohormones. That means a molecule that's not active, but it will be. Okay? So if something's a prohormone, then it's converted to, whatever, but we don't do much on that. But so they're talking about axon terminating the posterior pituitary. So they explain it, but it might be a little misleading. Then it goes through the function. Here's half-life of vasopressin, 16 to 24 minutes. Very short half-life. If anything has a half-life of minutes, basically you say that's very short, because some hormones have many hours of a half-life. But here's a case where Remember, 10 times half-life is gone, and so in 160 minutes, it's all gone. Doing 10 times 16. Pharmacokinetics, that's a great topic about, changes in things. Kinetics is things moving. Here's a great word, contraindication. Contralateral, remember contralateral? That's the opposite. So whenever you have a hormone or a drug, there's two things that you read about in that. Anybody if you get prostaglandin F2 alpha, and by the way, women should not touch that stuff, but if you have a bottle and you got it from the box, there's usually what's called a drug insert. It's a piece of paper that's got all the small writing on it. How many have seen those little pieces of paper? Yeah. So anyway, on that piece of paper, it might have side effects, but it has indications. Indications are what you could use this for, and then contraindications are, here's something you shouldn't use it for. Okay? And the reason I picked on PGF2 alpha is a very common desynchronized estrus in cattle. You used to have tons of it in a refrigerator, but it goes through your skin and it can cause abortion in pregnant that are women. Women that are pregnant, not pregnant that are women. Anyway, you know what I mean, if you're pregnant. Anyway, so be very careful when you're out there, because you should know many hormones can go through skin. And that's one way of applying hormones in some cases. It's called transdermal administration. Trans, T-R-A-N, dermal, across the dermis. So it's like the people that are trying to quit smoking, remember they have that patch, the nicotine patch or whatever, it gives them a low amount, it's supposed to stop your craving. That's transdermal administration of nicotine. And the bigger the patch, the more you get at a certain time. Anyway, so a function of vasopressin will, it's water balance. I'm going to just go through here real fast. 3 effects. You could write those down and never come back. Released within the brain. Sometimes things, that's the other thing you should be aware of. We might talk about 1 or 2 or 3 main effects, but some hormone might have 20 effects. And we don't talk about most of them unless you study them. Like analgesic effects. That means, you're decreased to pain. Who knows? Gene regulation, production of secretion. I'm just looking for words now. Okay, so the hypothalamus is at the base of the brain. It's all neural, and it makes a lot of different compounds. In fact, it's also your thermostat. A part of the hypothalamus helps you regulate your body temperature. But tell me, what brain tissue is above the hypothalamus? In fact, you're doing neuroanatomy.
Well, I don't know, cerebellum?
What's that? Oh, the cerebellum? No. What does hypo mean? Below. So the hypothalamus is below the thalamus. So there's a brain region called the thalamus. A little trick question, but just to say, sometimes it makes sense. Okay, so then we go on and on and on. Vasopressin during surgery and anesthesia. Vasopressin concentrate is used to measure surgical stress. That's interesting. Plasma vasopressin elevated by noxious stimuli. We're going to go on. I want to get to, okay. Now here's another thing. The other hormone that is released by the posterior pituitary is oxytocin. It's another compound that's used often in animal production. It causes uterine contractions. So it can help during parturition, that's birthing process. If somehow the animal has stopped contracting, the uterus has stopped contracting, oxytocin can help. It's got other effects. It causes milk letdown. When the cow comes into the milking parlor, they clean and massage the teats. There's a neural signal that goes to the brain, and the brain releases oxytocin. It travels in the blood down to the mammary tissue and causes milk letdown. Milk letdown is where all the little sacs that make milk contract and then sends milk out to the ducts so it can be harvested by the milking machine or the calf. Now here's a little interesting thing about these hormones. Notice I used the calf as an example, or the milker massaging. It's either the calf or the massager. But you know what? The sight of me can cause oxytocin to be released in some of my favorite milk cows. Why? Because if something is released by the brain, there is probably more than one mechanism, and it's called higher inputs. Just the sight of me and knowing I'm going to milk the cow causes her to release oxytocin. Or the sound of music in the milking parlor, she knows, because milk cow, she knows she's going to be milked, and that's releasing all that pressure in the mammary glands. She releases oxytocin without anybody touching her mammary glands. It's called higher input. Could be hearing, could be sight, and it could be smell. The smell of some milker that the cow, notice how I said the cow likes. Because if the cow didn't like any of that, it would release less oxytocin. So if there's a milker that's rough on the animals, that milker is going to get less milk out of a cow than somebody like me that talks to them and rubs their neck. Okay? I know that sounds funny, but it's true. Okay, so oxytocin is another hormone. Notice it's made of amino acids, and they've got the structure, how many are peptides consisting of, look at 9 amino acids. Just 9 amino acids strung together make these hormones. So they might be different in the cow or the sow. But there's a lot of what's called cross-reactivity too. That means one hormone from a cow, might work in a pig or a human, because maybe the key isn't perfect, but it does fit in and bring about effects, right? Okay. So then, I'm going to get to that adrenal gland. I mean, look at all these things. So like here, let's see, do they have oxytocin? Yeah, here it is. Here's oxytocin right here. Okay, vertebrates. There's the string of amino acids called oxytocin, and in most mammals, it's this structure. So if you're familiar with oxytocin, there's not a separate bottle for beef cattle or pigs or humans. It's all oxytocin, because all those animals have the same lock. So the key will fit into all of them. Kind of like a steroid, right? A steroid will fit in all the locks. But it's actually kind of unusual that like oxytocin will fit in all those locks, but it's so simple that it's easier to have consistency. But obviously there's differences. Okay, now, medical use, let's see. Are they going to talk about, so they're talking more about vasopressin than anything. Dizziness, all this stuff. See, there's contraindications. That means these are cases where you should not use it. It's contraindicated in the presence of hypersensitivity to beef or pork proteins. So that's interesting. Maybe what's in the bottle has some beef or pork product. Okay, recommended, cautious, interactions, okay. So let's go to that adrenal gland one. And this is a neat article. It's, I downloaded it, so it's on my website, but it's morphology of the adrenal gland. And whenever we'll be doing this in lab. Morphology is the study of structure. So adrenal glands are different shape in different animals. So you go on and you read this, but what I like about it, it's about a buffalo, but basically you can say, cows, buffaloes are similar. It's got a nice picture. Can't remember if this enlarges or not. And what's neat is A is the right adrenal gland and B is the left adrenal gland. So they don't have to look the same. In some animals, wouldn't that fool you if you think, this is what I'm used to. I'm used to this disc-like structure, and then if I made a cut here and then looked at the end, I would see cortex and medulla, and I could say, I got the adrenal gland. But look at this elongated one, rather interesting. Some are not that different. I think if I remember in the pig, they were both kind of elongated. But again, it's hard to find these things because they're not like this picture shows. If they were stuck on the kidney, that would be so sweet, because then you could find it. But it's always going to be either cranial or a little medial from from the kidney. And I think the horse is more medial. And if I remember right, the pig was more cranial from the kidney. So anyway, so this is a neat little article about adrenal glands. You know, it shows histology. And I know, you know, we won't be, you know, if you ever get a chance to take a histology class and making tissue, I did when I was working on my master's degree, I'd go out and collect all kinds of tissue from animals that were dead, you know, and do the staining and the processing. Kind of a fun technique, but time consuming. But if you just go through here, and then morphometric means putting some numbers on structure. And maybe sometime I'll show you some of the articles I did with boars. I did morphometric analysis on boar testes as they went through puberty, how things changed. So I castrated 40-day-old boars, 70-day-old boars, 100-day-old boars, 130-day-old boars, 160, I went up to 200 and something, and then I did histological study, and you can see how structures changed. And you can tell histologically if a boar has reached puberty or not. Anybody know how you can tell by histological examination of the testes if that animal has reached puberty or not? You know, puberty is the definition of the first time it releases gametes, which are sperm cells in the male. Anybody know that? If you see a histological section of a testis and the seminiferous tubules have a lumen, that's puberty. They're solid until puberty occurs. And when they have a lumen, that means they're starting to make sperm. If they're solid, they can't make sperm. And so in my boars, I could tell when they were going through puberty by not asking them, but doing the histological. But that's a great article, and you know, you're here to learn, and so you might say, well, that seems like a lot, but this isn't IU, this is Purdue. And I gotta think the best for you guys, right? I don't know what you're gonna be doing. 20 years from now, some of you guys are gonna be doing crazy, great things with animals. Okay, that's my story, and I'm sticking to it. You've got the videos to watch of the dissections, and by noon tomorrow, there's gonna be 2 practice paragraphs where I showed you. They'll say practice paragraph 1, practice 2, and it'll be fun because then Thursday you can say, what in the world were you thinking when you put down yellow? Have a great evening, everybody. Take care.
Question.
Good.
How might vitamin D deficiency affect other hormones? Or would it?
No, I don't think so because it tends to cause, it promotes calcium uptake from the gut. But I mean, you know, in a distant thing, I'm sure it can, but you know, right offhand, it's probably not going to affect other hormones directly that I can think of at the moment.
Okay.
Okay?
Mm-hmm. Thank you.
How can you tell when you're doing a necropsy or an autopsy? How can you tell that you have the adrenal gland?
Well, first of all, you're going to open it very carefully because the more you mess up, then if you start pulling things, you're done. So you carefully open it up. You know that they're going to be either cranial to the kidneys or a little medial from each one. And it might be in a blob of fat, I hate to say that, but you'll be, you know, you might have to take the kidney out carefully and examine that and then go to the other areas and might cut out some fat and then you might have to dissect that. My favorite thing to do is with blunt probes, you know, you take a blunt or 1 or 2 blunt probes and you actually pull things apart. You don't, you know, you don't cut, you pull and wait till you might see something. Because the less damage you do, the better. Because if you cut it, then it might look like a piece of muscle tissue. So, and they're not very big. So, but it'll be interesting because we have 3 animals to try to find it. So, and we have all semester, and we have over 100 animals to try to find. So, you know what I mean? Sure, we're going to find some. I just don't know who. Well, we should have a prize. You know what I mean? A candy bar for if you find the first one that finds an adrenal gland. Candy bar. Because I'm a little, you know, I don't know about the color preservation internally with these things. So we'll just have to see, but we'll take it step at a time. I'm sure we'll find an adrenal gland, I'm just not sure when.